You're not supposed to have giant cities in a tropical forest.
For decades, archaeologists focused on the end of the ancient Maya.
For centuries, the origins of the Maya appeared settled, filed neatly into textbooks and timelines. Then DNA disrupted the record. Genetic traces pulled from ancient remains began contradicting everything historians agreed on.
Most Maya kingdoms were simply abandoned to the forest.
But the start of Maya society holds as many mysteries as its fall.
Linking the Maya to movements far older and darker than expected. Each sequence tightened the picture, not clarifying it, but unsettling it.
This is the most important thing we're looking for, because it is the key to giving a date to the elements we have here.
What the data now reveals does not simply revise Maya history. It explains why the truth stayed buried for so long. Early human settlement of the Americas. Over 13,000 years ago, humans first crossed from Asia into the Americas via the Beringia land bridge. These early migrants moved south in multiple waves, spreading across North and Central America and adapting to new environments. By 10,000 years ago, Mesoamerica was home to genetically diverse hunter-gatherer communities. Archaeological evidence across Mexico, Belize, and Guatemala, including stone tools, food remains, and early habitation layers, shows continuous settlement rather than population replacement. These communities were not temporary camps. They had deep local roots and the knowledge to manage plants, seasons, and landscapes. Ancient DNA now confirms this complexity. Genetic markers link indigenous peoples across the Americas, but also reveal clear regional differences shaped by isolation, climate, and time. The Maya did not appear suddenly, nor did they descend from a single group. Their ancestors were part of a broader human presence that had already diversified over thousands of years. By the time agriculture emerged, this genetic foundation was firmly in place. Maize farming, permanent villages, and complex societies would build on this already varied ancestry. In other words, the Maya grew from populations whose genetic diversity was shaped long before pyramids rose or calendars tracked the stars. This rich, layered DNA set the stage for the astonishing discoveries researchers would later uncover, revealing how the Maya's origins are deeply tied to migrations stretching back to the last ice age. Belize DNA uncovers an unknown migration. Everything changed once scientists began extracting DNA from ancient bones in the Maya mountains of Belize. At several rock shelter sites, archaeologists uncovered 25 human burials dating from roughly 9,600 to 3,700 BCE. These were not ceremonial tombs or elite graves. They were ordinary people, buried across thousands of years, preserved just well enough to answer questions archaeologists had argued over for decades. The results challenged everything researchers thought they knew about Maya origins. The research, supported by the United States National Science Foundation, focused on ancient DNA recovered from these remains. What the results showed caught many scholars off guard. Around 5,600 years ago, the genetic makeup of people living in the region changed sharply. This was not gradual mixing or slow drift. It pointed to a major arrival of new people moving north from Central America. Genetic comparisons revealed that this incoming group was closely related to present-day Chibchan-speaking populations. These communities today live from southern Costa Rica through Panama and into Colombia. The link was clear enough that researchers could quantify it. More than half of the ancestry of later Maya populations traced back to this southern migration. That figure alone overturned long-standing models that treated early Maya ancestry as mostly local. What makes this discovery even more important is the timing. The arrival of these southern migrants aligns closely with clear changes in daily life. Around this period, archaeological evidence shows a rapid rise in maize consumption. Isotopic analysis of human teeth confirms that corn became a dominant food source shortly after this migration began. This strongly supports the idea that people were not just moving through the region. They were settling, farming, and reshaping the population. The lead genetic analysis involved researchers such as David Reich from Harvard University, working alongside regional archaeologists. Their conclusion was direct. This was not a minor influence or cultural borrowing. It was a population-level change that redefined who would later become the Maya. Instead of a single ancestral line growing in isolation, the evidence points to replacement through integration. New families arrived, mixed with local groups, and within generations became indistinguishable in the archaeological record. The Maya did not descend from one ancient community. They emerged from a merger that was far larger and more consequential than previously accepted. This discovery forces a harder look at how civilizations form. The roots of the Maya were not static or sealed. They were shaped by movement, connection, and large-scale human arrival that rewrote the genetic map of the region long before cities ever rose. Agriculture drives genetic change. Once the southern migrants settled into the Maya region, daily life began to change in measurable ways. Archaeological evidence shows that this period aligns closely with the arrival of fully domesticated crops, most notably maize, manioc, and chili peppers. According to research supported by the United States National Science Foundation, these were not experimental foods or occasional supplements. They became central to survival. Maize, in particular, tells the story clearly. Isotopic analysis of human remains shows a sharp rise in maize consumption between roughly 5,600 and 4,000 years ago. Before this point, diets were mixed and flexible. Afterward, corn became dominant. This was not a slow adjustment. It reflects a population that already knew how to grow, process, and depend on this crop. The timing lines up closely with the genetic turnover identified in the Belize rock shelter DNA. Manioc, also known as cassava, adds another piece to the picture. This crop requires specific knowledge to cultivate and prepare safely, as it can be toxic if processed incorrectly. Its appearance alongside maize suggests that these incoming groups brought established agricultural systems with them, not just seeds. Chili peppers followed the same pattern, reinforcing the idea of a complete food culture arriving intact. Geneticist David Reich of Harvard University has been direct about the implications. Based on genome-wide analysis, more than 50% of modern Maya ancestry traces back to this southern movement. That level of contribution is not compatible with minor contact or trade. It points to population growth driven by farming efficiency, food security, and settlement expansion. Agriculture changed more than diets. It reshaped population density, land use, and social organization. Farming allowed larger communities to remain in one place, raising birth rates and strengthening family networks. Over time, these communities absorbed remaining local groups, blending traditions while maintaining a strong genetic signal. What makes this period especially important is how tightly genetics and farming move together. The data does not show crops spreading first, with people following later. The people and the agriculture arrive as a package. This challenges older theories that framed early Maya development as a purely local invention inspired by distant neighbors. Instead, the evidence supports a model driven by migration, integration, and agricultural advantage. These southern groups did not replace culture. They merged into it, reshaping it from the inside. The Maya world that followed was built on this foundation, where farming knowledge and genetic ancestry were deeply linked from the very beginning. This moment marks a turning point. The Maya were no longer just adapting to their environment. They were actively redesigning it, guided by people whose arrival permanently altered the biological and agricultural future of the region. Continuity and diversity in Maya maternal lineages. As farming spread and populations grew, another question came into focus. Did these changes erase earlier communities, or did ancestry carry forward through generations? Mitochondrial DNA offers a clear way to answer that. Mitochondrial DNA, which passes only through mothers, shows that while overall genetic makeup changed dramatically, some maternal lineages from earlier populations persisted. Studies published in PubMed comparing ancient remains with living Maya populations show strong continuity. Both groups share the same core mitochondrial haplogroups A, B, C, and D. These haplogroups are common across indigenous populations of the Americas, but their specific frequencies inside the Maya region tell a more precise story. Rather than breaking apart, maternal lineages persisted even as populations expanded and mixed. Haplogroups A and C stand out in particular. High frequencies of both appear consistently in ancient samples and modern communities across the Yucatan Peninsula, Chiapas, and Quintana Roo. This pattern indicates that many maternal lines remained rooted in the region for thousands of years. Women were not replaced on a large scale. They stayed, raised families, and passed their ancestry forward through periods of change. Older theories often assumed early Maya groups developed in isolation, cut off from surrounding regions once farming took hold. The genetic evidence does not support that view. Instead, it points to long-term interaction across Mesoamerica, with movement, marriage, and exchange linking communities over time. Gene flow did not erase identity, it strengthened it. The maternal data show that even as new people entered the region and agriculture reshaped daily life, local lineages continued alongside newcomers. Over generations, these groups blended without severing maternal continuity. This kind of integration explains why Maya populations could grow rapidly while still maintaining deep regional roots. Geography also plays a role. Coastal and inland sites show slightly different haplogroup frequencies, suggesting regular contact between communities rather than rigid boundaries. Rivers, trade routes, and seasonal movement likely helped maintain these connections. The result was a population network that stayed connected while remaining diverse. Classic Maya genetic data from Copan. As the Maya world moved into the early Common Era, genetics began to track political growth in real time. One of the clearest windows into this period comes from Copan, a major classic Maya city located in present-day western Honduras. Between about 250 and 900 CE, Copan grew from a modest settlement into a powerful regional capital, complete with dynastic rulers, monumental architecture, and far-reaching alliances. DNA recovered from this site helps explain how that transformation happened. Researchers analyzed genetic material from 7 individuals buried at Copan, with remains dating across the early to middle Classic period. According to studies indexed in PubMed, these individuals share clear genetic continuity with earlier populations identified in Belize. This confirms that the people shaping classic Maya cities were not newcomers disconnected from earlier generations. At the same time, the data reveals something else. Genetic modeling detected approximately 6% gene flow from highland Mexican populations into Copan during this era. This finding, reported through phys.org summaries of the research, aligns closely with what archaeologists already suspected from inscriptions and artifacts. Copan was not isolated. It was connected to distant regions through political ties, trade networks, and elite alliances. What makes this evidence especially valuable is who these individuals were. The sampled remains include people believed to represent both elite and non-elite status. Their genetic profiles show a mix of maternal and paternal lineages rather than a single dominant family line. This challenges older assumptions that classic Maya cities were built by tightly closed ruling groups, reproducing within narrow bloodlines. Instead, Copan appears to have functioned as an open political center that absorbed people from different backgrounds. Some arrivals likely came through marriage alliances. Others may have moved as skilled specialists, scribes, or administrators. Over generations, these individuals became part of the city's core population without erasing local ancestry. This genetic blending mirrors what is seen in Copan's material culture. Architectural styles show influence from central Mexico. Writing systems evolve. Royal titles reference foreign origins. DNA now confirms that these connections were not symbolic. They involved real people becoming part of the population. Importantly, the 6% gene flow does not point to invasion or replacement. It reflects steady integration across centuries. Local ancestry remained dominant while outside lineages added complexity. This balance helps explain how Maya cities could adopt new ideas without losing continuity. The Copan data forces a reassessment of classic Maya identity. These cities were not built by static populations locked in place. They were shaped by movement, negotiation, and selective integration. Power did not come from genetic purity. It came from a connection. By the time Copan reached its peak, its people carried ancestry stretching back to early Belize communities, combined with smaller contributions from distant regions. Population growth and change. By the early 8th century, the Maya world reached its high point in numbers. Genomic modeling published and summarized by Live Science estimates that population levels across the lowlands peaked around 730 CE. This growth was not accidental. It followed centuries of agricultural refinement, with maize at the center. Irrigation systems, terracing, and intensive land use allowed cities to support dense populations on a scale unseen before in the region. But the same genetic tools that reveal this peak also record what came next. Around 750 CE, researchers observed a sharp decline in effective population size. This does not mean millions disappeared overnight. In genetics, effective population size reflects how many people are contributing to future generations. When that number drops quickly, it signals widespread disruption. In the Maya case, it lines up closely with the breakdown of classic period political systems. Major centers such as Tikal, Copan, and Calakmul experienced declining monument construction, abandoned palaces, and reduced elite activity during this period. Climate data adds another layer. Multiple studies link the 8th and 9th centuries to prolonged drought cycles in the Maya lowlands. Agricultural systems that once supported growth became harder to maintain under stress. DNA evidence supports this picture of contraction rather than disappearance. According to analyses cited by Journals of India, genetic continuity persists across this period. The Maya people did not vanish. Instead, populations are redistributed. Some communities moved toward more reliable water sources. Others broke into smaller settlements that were easier to sustain. The genetic record shows survival through adaptation, not extinction. This matters because it reframes one of history's most misunderstood moments. For decades, the classic Maya collapse was portrayed as sudden and total. Genomic data rejects that narrative. The decline was real, but it unfolded through demographic thinning and regional reorganization. Families continued. Lineages endured. What collapsed were the systems that held large cities together. There is also evidence that social change followed population pressure. As the central authority weakened, power likely shifted away from dynastic elites toward local leadership. This transition would reduce long-distance coordination but strengthen regional autonomy. Genetics reflects this through reduced mixing between distant centers and more localized reproduction patterns. By the end of the 9th century, the Maya world looked very different, but it was still very much alive. People spoke related languages, farmed familiar crops, and carried ancestry that stretched back thousands of years. The population peak and decline were not the end. They were a turning point. Despite these pressures, genetic evidence confirms that Maya lineages endured. DNA shows descendants of classic Maya populations remained in the region. The collapse transformed political systems and urban centers, but it did not break genetic continuity. People redistributed across the landscape, adapted to new conditions, and carried their ancestry forward. Gender, kinship, and burial DNA. When researchers turned their attention to Chichen Itza, they were not looking for kings or conquest stories. They were looking at human remains. What they uncovered added an entirely new dimension to how Maya society functioned. Genome sequencing published in Nature analyzed individuals from mass burials dated between roughly 500 and 900 CE, a period when Chichen Itza was rising as a major political and ceremonial center. The genetic results were striking. Many of the buried individuals were closely related males. In several cases, researchers identified brothers. In one burial cluster, the DNA revealed identical twins. This was not a random cross-section of the population. The odds of finding such close kin relationships by chance are extremely low. Instead, the data points to deliberate selection. These findings raise important questions about who these individuals were and why they were buried together. Archaeological context shows that many of the remains came from ritual settings rather than ordinary graves. This suggests that family ties played a role in religious or ceremonial practices. Rather than being anonymous victims or unrelated participants, these men appear to have been chosen because of who they were within their families and lineages. The gender pattern matters as well. The majority of individuals in these clusters were male, which aligns with iconographic and textual evidence that certain public rituals were dominated by men. DNA now supports the idea that male kin groups may have held specific ceremonial roles, possibly tied to status, obligation, or lineage-based authority. What makes this discovery especially revealing is how it changes assumptions about Maya elites. Older models often imagined ruling families as isolated bloodlines ruling from the top down. The genetic data from Chichen Itza suggests something more complex. Elite identity may not have been limited to a single royal household. Instead, it may have involved networks of related families whose members participated in ritual life together. DNA also helps explain why these burials look different from typical Maya graves. The clustering of close male relatives points to structured practices rather than chaotic events. These were planned acts embedded in social rules that valued kinship as much as power. This research shows how genetics can uncover social organization that texts never recorded. Inscriptions tell us about rulers and dates. DNA reveals who stood beside them and how families were woven into public life. At Chichen Itza, ancestry was not just a biological record. It shaped identity, duty, and participation in the city's most important moments. The genome data does not simply tell us who these people were related to. It shows how Maya society organized itself around family ties in ways historians are only beginning to understand. Modern Maya and ancient connections. DNA research has fundamentally changed how we understand Maya origins. The Maya did not arise from a single isolated population. They were shaped by multiple migrations spanning thousands of years. Around 5,600 years ago, southern migrants from Central America contributed over half of later Maya ancestry, bringing agricultural knowledge that fueled population growth. During the Classic period, highland Mexican gene flow added further complexity. Despite these layers of movement and integration, genetic continuity persisted, linking modern Maya populations directly to their ancient ancestors. The Maya were not born from isolation, but from connection, adaptation, and the gradual layering of populations across millennia. New genetic paradigm: complexity over simplicity. Recent DNA research supported by the United States National Science Foundation paints a picture of multiple migrations, ongoing gene flow, and repeated cultural exchange. The Maya did not appear from a single isolated population. They were shaped by movement, interaction, and the gradual layering of new communities on existing ones over thousands of years. Each wave added genetic and cultural variation that would influence later generations. Genomic studies reveal that southern migrants arriving around 5,600 years ago contributed significantly to the ancestry of the Maya, while earlier populations from Northeast Asia via Beringia provided the foundational lineages that settled Mesoamerica. This mixing of groups brought agricultural knowledge, particularly maize, manioc, and chili peppers, which allowed populations to expand and cities to grow. Far from being static, the Maya were adapting, experimenting, and integrating innovations from neighbors as they moved and merged across the region. Evidence also points to ongoing gene flow during the Classic period. Analysis of remains from Copan shows a small but measurable influx of highland Mexican ancestry between 250 and 900 CE. This genetic input aligns with archaeological signs of political alliances, trade, and shared cultural practices. DNA confirms that these interactions were not superficial. They became embedded in the population, producing a network of communities tied together through both bloodlines and shared knowledge. By the post-Classic period, Maya populations had absorbed these influences while retaining deep connections to their ancestral roots. Genetic continuity demonstrates that even during periods of social stress, climate challenges, and urban decline, populations endured. Lineages persisted, and communities adapted rather than disappearing. Modern Maya populations carry this legacy today, maintaining strong indigenous ancestry alongside minor European and African contributions from the colonial era. This new paradigm overturns decades of assumptions about Maya formation. Ancient DNA shows that civilization did not emerge from a single place or simple process. Instead, the Maya developed through dynamic interregional movements, sustained agricultural innovation, and continuous adaptation to environmental and social pressures. Genetic evidence now complements archaeological and linguistic data, creating a multidimensional understanding of how the Maya formed, flourished, and persisted over thousands of years. The Maya were not a singular invention of time or place. They were the product of interconnected histories, migrations, and innovations, demonstrating that complexity, rather than simplicity, defines the origins and endurance of one of the most advanced civilizations of the ancient world. Thanks for watching. See you on the next one.